Anaiah, and my project is about the manipulation of natural killer cell phenotype and function using fibroblasts. So in the grand scheme of things, my project is just about the immune system, which is obviously a very relevant area of study right now, being that we live in the middle of a pandemic and people with immune deficiencies are less likely to recover and are more susceptible to diseases like the coronavirus. So that's why it's really important that we learn as much as we can about the immune system, being that it is the forefront of our health. My project specifically investigates a specific a specific type of immune cell called a natural killer cell, which is a type of innate immune cell, meaning that it's kind of one of the first runners up whenever you have an infection or any disease in your body. And it essentially comes in two main subsets, CD56 bright and CD56 dim. So CD56 bright natural killer cells have more CD56 ligands expressed on the cell surface. They are more known for immunosuppression, so turning down your immune system, and they don't have much cytotoxicity, so that can be expressed in granzymes, which is just a molecule that kind of attacks and kills other cells. So they're not very good at killing other cells because they're more involved with immune suppression. On the other hand, CD56 bright natural killer cells are involved in immune activation. They have less CD56, but they have more granzymes, more cytotoxicity, and are better at killing other cells. However, there's a lot of controversy about the cell lineages of these two subsets, how they interact with each other, their functions. There's just a lot more to be known about how these cells interact in the body, and that could obviously contribute a lot of information about the immune system and our health. For example, if you're able to understand more about natural killer cells that are CD56 bright, you could maybe fine-tune their function so that you can upregulate CD56 bright expression when you have diseases like autoimmune diseases where your body is kind of attacking itself. You can induce more immunosuppressive cells to appear in a specific area in the body. On the other hand, if you have a cancer or coronavirus or any other disease or infection, you would be able to learn more about CD56 dim natural killer cells and maybe upregulate their production in another part of the body so that you're able to kind of fine-tune your immune system and help it do its job better. So kind of a clue in our study of these two subsets is the fibroblast, which is believed to kind of contribute to differentiation between CD56 bright and CD56 dim cells. So people believe, or research shows that these fibroblasts may contribute to converting a CD56 bright cell to a CD56 dim cell, or maybe there's some fluid interaction with different fibroblasts and different versions of natural killer cells, or maybe there's something else going on that we don't yet understand. My project really investigates what happens when you co-culture CD56 bright natural killer cells and fibroblasts, different types of fibroblasts, and how we can use the information that we get from this data to kind of further our knowledge about the immune system and about these cells. I did a lot of different facets of this study. I studied these interactions with activated and resting natural killer cells. I studied them with primary culture and cell line culture fibroblasts, with adherent natural killer cells that were adhered to the fibroblasts and natural killer cells that were floating around, natural killer cells that were given supername, and natural killer cells that were allowed cell-to-cell contact. So there was a lot of different facets that I took in this study, which is why I kind of have a lot of data here and got a lot of different results that sometimes conflicted with each other, but in the end, I did what I set out to do. I learned more about these amazing cells and contributed to furthering our knowledge of these cells so that we may better be able to treat diseases. Thank you.